My dear students of 4th SEM, as you are locked down in your house due to coronavirus outbreak, the college authority has taken an initiative to teach through online classes. As a part of this endeavor, I am going to discuss with you all the split gene concept, which is included in the syllabus of Core Course 10. As we all know, and as you can see from the slide, the DNA transcribes the RNA. The genetic code is stored in DNA in the form of deoxyribonucleotides, which is copied into RNA as ribonucleotides. This RNA directs the synthesis of polypeptides, which is made up of amino acids. The first step in the expression of genes is known as transcription, whereby an RNA complementary to DNA is being formed. It occurs inside the nucleus. The second step, that is translation, where the information contained in the mRNA is converted into the language of amino acids present in polypeptides, is called translation, and it occurs inside the cytoplasm. Then the next slide will show a brief concept about the split genes. Before that, I want to say that the transcription in eukaryotes is more complicated than that in prokaryotes. In 1977, an exciting discovery was made in the history of molecular genetics. Three scientists, namely Susan Bargett, Philip Sharp, and Richard Roberts, showed that genes of animal viruses contain internal nucleotide sequences that are not expressed. That is, they do not produce any amino acids in the protein they encode. These internal DNA sequences are present in the initial primary transcript but are removed before the mature transcript takes part in translation. Such nucleotide sequences are called intervening sequences, and the genes containing them are called split genes. Those DNA sequences that are not present in the final mRNA transcript are called introns, and those that are retained and expressed are called exons. Thus, the split gene consists of exons, introns, and again exons lying adjacent to each other. The exons are also known as essential or informative sequences. This is the exon, and introns are known also as non-essential, non-informative sequences. Splicing helps in cutting the introns and joining the exons into a final mRNA transcript. Now, the number of exons and introns varies considerably in different species. which I'll show you in the next slide. Reports of split genes have been obtained from chicken ova-albumin genes, beta-globin genes of rabbit, mouse insulin genes, from the ribosomal genes of Drosophila, and even from the tRNA genes of yeast. As you can see, the number of nucleotides contained vary in different organisms, that is, the number of nucleotides present in introns and exons varies considerably in different organisms. Next slide. From the gene, that is in the split gene, certain internal sequences are spliced off, as you can see, and this is one of the part of processing of the initial primary RNA transcript to the mature mRNA transcript. And during this process, two other things occur. One is addition of a 7-methyl guanosine cap at the 5' end and the addition of a polyadenyl tail at the 3' end. The latter two features which I have just mentioned are not included in the split gene, but still I have mentioned here to give a glimpse of what happens during the processing of RNA. Introns consist of a dinucleotide sequence. At the 5' end of exon-intron junction, GU is present, and at the 3' end, AG is present. This shows the non-template strand of DNA from which the transcript, the RNA transcript is being formed. So thymine is replaced by uracil, and hence it is GU and not GT in the transcript. In the plant system, introns have been reported from French beans. French beans contain a structural protein, fasciolin. The gene that encodes this protein contains 3 introns, while in other plants, a number of introns or multiple introns are present. The dinucleotide sequence which I have talked about with you earlier is consensus, that is present more or less in all organisms. The location of introns is same in different organisms as it has been found in mouse and even in soybean. The question arises, if introns are non-informative and non-essential, then what is its biological significance? What function they have? Do they have any function? Yes, they have certain functions. Introns are made of repetitive sequences, and at this intron site, recombination takes place, which leads to reshuffling of exons. Introns have originated early in evolution. Now, when we trace the evolution of land plants, it has been seen that the bryophytes have evolved from the algal system, and this is shown by the similarity of introns present in bryophytes and in algae like Chara and Coleochaete. Now, this is in all, which are a nutshell about the split gene, that is the structural organization of the split gene. I hope you all have understood. If there is any question, you all can always ask me or send an SMS. Thank you.